Hello and welcome to the mind map about osmoregulation and nitrogenous wastes. Okay, we're going to zoom on in. We'll go up here to the left-hand side where we're going to talk about nitrogenous wastes. All right, nitrogenous wastes are made in the liver by the deamination of proteins and amino acids. But this creates ammonia that is very, very, very toxic. Mammals are able to turn ammonia into urea by reacting with carbon dioxide inside the liver. Uric acid is able to be made by some organisms by the destruction of adenine and guanine, and uric acid has its advantages. All right, then we're going to talk about the three types of nitrogenous waste. There is ammonia that is very, very toxic, but is incredibly water soluble, so it's very easy to get rid of by animals that live in water. Next, we have urea, which has a medium toxicity and is also still relatively soluble in water. But then we also have uric acid that is the least toxic and not soluble in water and is therefore excreted as a paste. Right, here are the three nitrogenous wastes, and it shows you what sort of animals excrete them. First is ammonia. Here it is NH3. It is excreted by things that live in water. Next is urea, has 2 nitrogen atoms per molecule, and it's found in organisms that have water in some abundance. Last of all, we have uric acid. It has 4 nitrogen atoms per molecule, so it's very expensive to make, energy-wise, and it's excreted by reptiles and birds and insects and land snails. Okay, then we're going down to osmoconformers and osmoregulators. Now, an osmoconformer is an organism that keeps its body fluids isotonic with that of the environment. It saves a lot of energy on osmoregulation because it doesn't have to do it, but it lives in a narrow range of salinities. The external environment remains constant, and therefore the osmolarity of, say, the octopus remains constant as well. Within the osmoconformers, we've got a subgroup called euryhaline, and the euryhaline organisms can survive a wide range of osmolarities by changing their own bodily fluid concentration. So technically, a bull shark is both an osmoregulator and a euryhaline individual. Bull sharks change the osmolarity of their tissues by either keeping or excreting urea. So bull sharks can survive in marine and brackish water and, to a certain extent, freshwater. Bull sharks, though, if they're going to go from marine to freshwater, they need to spend a month or two in brackish water to acclimatize. All right, next of all, we've got the osmoregulators. Now, osmoregulators use osmoregulation, that is negative feedback, and the osmoregulators are also defined as stenohaline, which is stenohaline, and the stenohalines monitor or maintain their osmolarity within a narrow range of salinities. All right, so first of all, we'll look at marine fish. Now, marine fish are exposed to the problem where the salt water is more hypertonic than the fish. So fish lose water through its gills via osmosis. Therefore, it needs to drink seawater to get the water, but now it needs to get rid of the salt. And it can excrete the salt through its gills using active transport. It has fewer nephrons, and therefore its urine is made mainly by the excretion of ions. It makes a small volume of concentrated urine, and the urine is also isotonic to the fish's blood. Next, we've got freshwater fish, and they have the opposite problem. The fish is more hypertonic than the water that it's living in, so water is going to rush into the fish through the gills using the process of osmosis. So these fish, the freshwater fish, don't need to drink water, but they do need to take in ions, as you lose ions whenever you urinate. So it takes in ions through the gills and in the food that it eats. Now, it's going to have a lot more water than a marine fish, so it needs to get rid of it. And so it has lots of nephrons in the kidney. All right, and we say that it makes its urine in a normal fashion, but it makes it in large volumes and very dilute. Now, over here, we've got a wonderful diagram you should all take note of that compares saltwater fish to freshwater fish. Okay, and if you ever get a question about osmoregulation in fish, this would be a lovely diagram to use as your plan. Okay, next of all, we'll rush up to here and we'll go through osmosis again. So osmosis is the net flow of water through a semi-permeable membrane from a higher concentration of water, which we call hypotonic, to a lower concentration of water, which we call hypertonic, along the osmotic gradient, which is the concentration gradient for water, and it's passive in that it requires no energy. Okay, if we look at the key terms again, osmolarity is the relative ratio of solute to solvent. So in this case, it's ions to water. And osmotic pressure is the ability of a solution to attract water. So if you are hypertonic, you will attract water. Therefore, a hypertonic solution will have a higher osmotic pressure. The word hypertonic means a higher solute concentration and a lower solvent concentration. The word hypotonic means a lower solute concentration and a higher solvent concentration. And isotonic means equal solute and solvent concentrations on either side of a membrane. Remember, the solvent in cells is water. We don't need to worry too much about the movement of water or the loss of water, the movement of water via osmosis in plants and animals and what they're called. That's mainly for year 11. So we'll go down to here for terrestrial organisms. And if you're a terrestrial organism, you're going to lose water a number of ways. You're going to lose it through your respiratory surfaces via evaporation. You're going to lose it as a liquid in your feces. You're going to lose it via evaporation in sweat. And you're also going to lose it in urine as a liquid. Okay, now the kidneys. The kidneys are the ones that make the urine and therefore decide how much water is going to be lost as urine. So kidneys filter the blood, and they filter the blood in humans about once every 5 minutes. The formation of the urine is in a structure called the nephron, and the nephron has a number of interesting features. In the proximal convoluted tubule, there is obligatory osmosis because in the proximal convoluted tubule, you've got the active uptake of ions, and therefore water will follow. That's what obligatory means. It has no choice. So the active uptake of ions creates a hypertonic environment inside the blood, and water must follow. Okay, then we've got the loop of Henle. The loop of Henle works back to front, sort of. Right, the ascending limb, which is the limb going upwards, reabsorbs sodium via an active process. And that amount of sodium creates a hypertonic environment in the middle of the loop, and water must follow again through an obligatory process of osmosis. That's why I say it works back to front, because the ascending limb dictates the amount of water reabsorbed via osmosis in the descending limb. Then we've got the collecting duct, and the collecting duct is susceptible to the hormone, antidiuretic hormone, and this allows facultative osmosis, which means it can be switched on or switched off via the addition of ADH, which puts in aquaporins into both the collecting duct and the distal convoluted tubule and allows for more water to be reabsorbed when the organism is dehydrated. Okay, now if we look down here at the specialized structures of different sorts of organisms, both animals and plants, we can see that some organisms have salt glands that squirt hypersaline solutions from inside the organism to outside the organism. Birds do it. The albatross here does it. It's got specialized glands here, the nasal salt glands. And if you find a video, you can actually see the albatross squirting out hypersaline solution from its nose. Iguanas do it as well, and so do mangroves, but it's a little bit less impressive when mangroves do it. Okay, they can also get all of their water from food. We call this metabolic water, and organisms like the desert hopping mouse do it. Some organisms also have a very long loop of Henle, and the best example of this is the desert hopping mouse that has the longest loop of Henle for its body size. And the longer the loop of Henle, the higher the ability of the kidney to reabsorb water. Some organisms rely upon fog condensation on their shells, and then they drink the fog as it turns into water. Some organisms manipulate the solute concentration of their bodily fluids, including bull sharks. We also know that mangroves do this as well. We know that some organisms lose ammonia directly through their gills, i.e. fish. We know that some organisms prevent salt from entering their root. Mangroves have a waxy membrane that is impermeable to salt but lets water through. You can store salt for removal, And we know mangroves do this as well. They store salt in vacuoles and also in bladders and salt glands. And this takes the salt either out of their body or out of the cytoplasm. Some organisms also pump salt directly out of their roots via active transport, and mangroves are a great example of this. Okay, so we'll zoom on out, and thank you for listening, and goodbye.